Welcome to the video lesson on rods and cones. The rods and cones, trichromatic theory, and opponent process theory, all of the class notes are on one PDF file. I've broken it up into 3 video lessons. Now in this picture, we have the cat, and it traces the pathway of vision. It will travel through the cornea, through the pupil, through the lens, and the image is actually inverted onto the back of the eye. It hits the retina, inverted, our brain corrects it. The retina is this thin membrane at the back of the eyeball containing rods and cones. And in the picture down below, we have the rods that are depicted in light blue, and they're rod-shaped, and cones that are cone-shaped and a magenta color. Now the rods and cones have different functions, and in your book it kind of describes them. It goes back and forth between rods and cones, and if you can talk about them all at once, hopefully you'll be able to remember all of the important information of rods at once, and then all the important information of cones at once. And part of the homework assignment that I have is to put the important pieces of information, some of the important pieces of information, in 2 different columns. So we have rods and cones in the back of the eye, and one of the curious things, or at least I find curious, is that the part that's sensitive to light is on the very back end of the rod and cone. So this pink area with a dot in it represents the part of the rod and cone that's sensitive to light. So the light has to travel through the cornea, through the pupil, through the lens, through a fluid in the eye, through these ganglion cells, through the bipolar cells, through the actual body of the rod and cone, and then it hits the light-sensitive part. Excuse me. Cones, they estimated there's 6 to 7 million of them. Depending on what book you use, 6 to 7 million, somewhere in that area. Exact number is not overly important as long as it's in that 6 to 7 million region. The cones generally have 2 specialized functions. One is visual acuity. I prefer to use the word perception of fine detail. Many students don't use that word acuity, so they have a little bit of problem with it. So I prefer the word fine detail. If you look up above, the fovea is the highest point of visual acuity, being able to see fine detail, and that's because cones are concentrated there. So cones are important for fine detail, and the fovea is the center of our vision. So when we look at something with a center of our vision, we're basically looking at our fovea, we're able to see fine detail. The other function of the cones is color perception. Cones, as we'll see, is one of the things that is responsible for our ability to see color. Now going back up to the picture, up above, fovea. Cones are concentrated in the fovea. There are no rods there. And outside of the fovea, cones are scattered throughout the retina. So we have cones all over the place, but they're really concentrated in the fovea. The other ones, the rods, these long cylindrical cells, estimate 125 million. I've seen some books put 120 million, somewhere in that region. Also looking at the picture, we can see that there are more rods than there are cones. So if you're a visual person, you can use this picture to help you remember which one has more. Since there's more rods in the picture, they're the 125 million. The cones would be the 7 million. The rods are responsible for seeing dim light. And as I mentioned before, there are no rods in the fovea. So when we look at things with the center of our vision, we're using our cones and not our rods. If we want to see something in dim light, we can't look directly at it. And I've been told when sailors had to navigate by the stars, if they wanted to see a star, they would look with their peripheral vision because they would be using their rods. So it'd be a lot easier to find the star when they didn't look directly at it. So these rods are spread out all over the retina, none in the fovea. Now I want to go down to the next picture, the blind spot. Now, this over on the left-hand side, it's the same picture that we had before. We have the rods and cones, the light-sensitive part on the back side of the eye. Once it detects light, the signal travels forward, going left on the picture, forward through the eye, into some nerve endings and exits through the optic nerve. And there's a place called the optic disc where there are no rods and cones. So we don't perceive anything in those blind spots. Now we don't notice the blind spot in everyday usage. We don't see these big empty gaps when we walk around in the world, partially because our eyes move a little bit and we have two eyes and our brain fills in the missing pieces of information. So if you can print out these class notes, or you have the book has the spider with a can of Raid. I prefer this one from an old Myers textbook with a car. But what you're supposed to do is if you print it out, put the black dot about a foot away from your right eye, and using only your right eye, closing the left, stare at the black dot and move the image back and forth, move the paper back and forth until the card disappears. For me, it's about 8 inches. And when that card disappears, all it's filled in is this kind of ivory colored background. The brain fills it in. There's not this dark gap where there's nothing there. The brain kind of fills in that information. Now the reason why I like the car one instead of the spider one in your book is that if you're driving and only looking at the world with one eye, which doesn't happen often, but if we are turning left or right, sometimes we do that really quickly, and in fact we're only using one eye when we're looking quickly to the left or to the right. And I've had a couple of times where when I did that, a car happened to have been in my blind spot, and then all of a sudden a car comes out of the middle of nowhere. So that's why I like this image. It could be a car, could be a motorcyclist, could be a pedestrian, could be a dog. So something can hide in that blind spot. And the brain fills in the missing pieces of information. Now to illustrate how the brain fills in information with what we expect, take a look at this image. The first time I did it, I read it wrong. Some of you may be able to figure out what's wrong with it. Some of you may have a difficult time. And if you have a difficult time, it's just the brain filling in and interpreting the world around it with what it knows. If you're still having a difficult time, read each one of the 4 lines slowly. Because if we go fast, it's very easy for us to read this wrong. This concludes the first video lesson for rods and cones. The next 2 will be on color perception. Thank you.